Hi, hey, hello, and welcome back to another episode of the Bovine Podcast, brought to you by Alberta beef producers. I'm your host, Kara Mastel, and as always, I'm happy to be here. Recently, ABP hosted the Beef Research Showcase at Lakeland College in Vermilion, Alberta. This event highlighted the latest advancements in beef and forage research, featuring presentations on topics such as soil microbiomes, herd health, intercropping, and pasture productivity, and of course, much more. Attendees had the opportunity to engage with experts, explore innovative research findings, and discuss practical applications for improving beef production and sustainability. Today, we're going to dive into one of the research topics that was discussed, soil microbiomes, with Dr. John Bennett, who's an associate professor at the University of Saskatchewan. John is here with us today. John, first off, thanks for joining me on the Bovine. Rather than me dive into who you are and your background, would you like to tell us a bit about yourself?
I'm John Bennett. I'm an associate professor of Plant Ecology at the University of Saskatchewan, working a lot on the ecology and management of rangeland and forage systems. A lot of our work is on plant soil microbe interactions, and it kind of stems from the work I did started during my PhD at the University of Alberta, where I worked on plant ecology and plant-microbe interactions at the Kinsella Research Ranch. And since then, I've done, postdocs in both British Columbia and in Europe, kind of rounding out my skill sets in that area. And so,
Quite the resume of different places you've been to then.
I've, I did undergrad in Nova Scotia, master's in Ontario, PhD in Alberta. So I'm coast to coast really in Canada here.
So are you originally from the East Coast, or what brought you out west?
I am, yeah. Born in Newfoundland, but I haven't been there I've been there, but I haven't lived there for more than 30 years.
So now Western Canada's home.
It is actually. My dad was with the RCMP and he got transferred to Prince Albert for high school for me. And so I left a little bit after that for some of my schooling, but yeah, I think I've spent more time in Western Canada than anywhere else at this point. And actually, I think of any city, I think I lived in Edmonton the longest. I like Edmonton, so.
No, it's a good place. So what brought you on this career path? Well, I'm always curious. Well, what kind of got you there?
Yeah, sure. Yeah. So in my undergrad, I actually had a passion for insects. And so I wanted to work on insects. And so I went and I did a master's degree on biological control of insects in tomato greenhouse production. And what I realized when I was working on that is that I couldn't understand anything that was happening with the insects without understanding the plants. And so then I did a master's in plant ecology and then kind of dabbled a little bit in plant microbe interactions there. And that's it's a very complicated system and I kind of like complicated things. So I've been dabbling in it since.
Yeah, it's amazing how much goes on that we can't see without a microscope, and even then sometimes is very tricky.
Oh yeah, microscope doesn't even get us close to like, yeah, I get motion sick if I use a microscope for too long. So we don't use them all that much in my lab. My students do sometimes, but for me, it's mostly, we've moved mostly to DNA based assessment for the microbial stuff. And so, because there's no real way to identify them all that confidently with a microscope.
Wow. Okay, so you recently spoke at the Beef Research Showcase at Lakeland College. You spoke about soil microbiome and all the things that go along with that. Do you want to talk a bit about what your presentation was about and maybe go into why the soil microbiome is important for forage yields?
Yeah, and so one of the things, like I mentioned, we've been working on plant microbe interactions in the range of forage systems for a while now. And one I talked about at Lakeline was a bit of a summary of some of the work we're doing around trying to kind of optimize plant microbe interactions in some of these systems, particularly in forage systems. It's a little bit more challenging to do in rangelands because obviously you're not going to go reseed a rangeland. And so you have the number of levers you can pull are more difficult, but we are working a little bit on some grazing related things, but um, not a lot you can really manipulate there otherwise. And but so we work a lot of that's around forage systems, and there's two real ways that we've been trying to address optimizing plant microbe interactions in forage systems. That's about part of that's around inoculum development, or at least kind of trying to find ways to develop inoculums. And the other is around trying to figure out what plants work best with the microbes so we can kind of maximize the benefit you get from that. And so I guess like the complexity of it. And so, we often think about microbes. Well, we don't not to think about microbes, but when we do, there's lots of different things that they can do. Some of them are beneficial, some of them are antagonistic or negative. So things like pathogens. And then there's some that just kind of do their job and cycle nutrients through those systems. And so a lot of the work we do is really focused on that kind of, at least in the forage systems, is around kind of maximizing the amount of the benefit that plants get from those potentially beneficial microbes and minimizing the negative effects of pathogens. And so I think it can be an important way of kind of leveraging something that's kind of already there or something that we put into that system to kind of increase plant growth and maximize the benefits that we're getting from them for forest systems, that is.
Absolutely. And you're saying, okay, maybe we don't always think about microbes, but why should the general producer actually be paying attention to microbes and maybe looking at some of the research that's being done? And is it all come down to maximizing that yield?
Well, there's lots of things microbes can do, but certainly, that from a producer's bottom line, I think maximizing the yield and minimizing inputs is probably the two biggest things financially that might be beneficial for the average producer. Like you don't, if you can have a plant that uses like an alfalfa plant, for example, that benefits more from certain microbes. Like, so we work a lot on arbuscular mycorrhizal fungi, which are these fungi that live inside the roots of the plant host and they can't really live outside of those things. And basically, they act oftentimes as an extension of the root system and they forage for nutrients in those soils and those nutrients get exchanged with the plant in terms like so that they can't derive their own carbon or their own sugars or fats or anything like that, and so the plant gives them that. And they the most common transaction anyway or most common interaction is that they give phosphorus or some other micronutrients to the plant in exchange for those um, you know products of photosynthesis, so sugars and those fats. And so if you can manage the system to maximize those benefits, it really would, really should really cut down on like fertilizer costs if you're actually able to use microbes to mine the existing, the kind of inaccessible stores of some less mobile nutrients like phosphorus within that system.
And how do you get your toes wet with that? Like, how do you actually optimize these interactions and help them improve the plant soil interactions?
That's a fantastic question. And we have about a half dozen different projects trying to tackle it from different directions. Um, like from a producer perspective, it's a little challenging right now. I think, we've done a lot, and especially as we've bred a lot of crop plants and things like that. Often we're breeding plants that are most productive in kind of like high input systems. And that frequently selects for plants that aren't really capable of, they're more reliant on kind of nutrient like fertilizer inputs than they are on being able to scavenge for nutrients in a like in a less fertile environment. And so oftentimes that selects for plants that don't actually have these beneficial interactions with soil fungi. And what one of the avenues that we're trying to take right now is just identifying existing varieties that, where that selection for or selection against, I guess, like beneficial microbes has been like, well, that either hasn't happened or it's been less intense. And so you've still got varieties out there that can benefit from these things. It's just I don't think we know which ones they are. And so I think the first step for us has really been trying to identify which of these varieties can kind of like leverage a microbiome in order to kind of maximize their own growth. And, so I think ideally within the next couple of years, we've got recommendations we can make on that. We still, a lot of our work has been in greenhouse settings. And so that's, we're really we're pushing now to take this stuff out into the field, because obviously a plant growing in a greenhouse isn't exposed to the same stresses that a plant grown out in a pasture would be, especially, you can't just turn on the tap out in the pasture. And so you don't know kind of whether those conditions are far less regular or amenable to kind of like plant production. And so we need to kind of see whether or not the principles that we're finding in our more controlled studies are still kind of, if we can leverage them under a field environment.
Is there plans to do large scale research there?
We are, yeah. So not necessarily focused purely on mycorrhizas, but we've got a few, we're trying to round up some money right now to do a few of these things. We have been we've done a fair bit of greenhouse work, for example, on trying to identify alfalfa cultivars that do well with the microbiomes from old pasture soils. And so we inoculated 21 different seed sources with microbes from 36 different locations spread across 12 pastures through all the soil zones of Saskatchewan. And, some of those cultivars did relatively well. On average, it probably increased their growth by 30% or more when inoculated with those soils from the old fields. And some of them, like their growth dropped off by 20-30%. So you've got this huge range in responses to how they grow when you inoculate them with the microbes from those old fields. And so right now we're trying to, get the money together to be able to take that out into the field. And the plan is to basically find, interested producers that might want us to come take some samples from their field and then grow a bunch of different stuff in that soil the year before, and the next year come out and seed the best performing ones into fields to see which cultivars or potentially other forage legume species are going to really respond positively to that particular field, with the ultimate goal for us being able to develop some sort of either greenhouse assay or soil test that we could use to like really improve cultivar selection for people, at least in the rejuvenation scenario. But I can't see why it wouldn't work under any other conditions as well. It's just not really been our focus, because I think those old pastures that have historically had alfalfa are probably already have a lot of the same potential disease causing organisms within those soils. And so that's really where I think it's probably going to be, more critical to success than if you, seeded it to barley for a few years and then decide to turn it into perennial pasture. In that scenario, you might not have the same density of alfalfa specific pathogens than you would in kind of like a rejuvenation or renovation or whatever you want to call it.
Yeah, absolutely. That makes sense. It's basic crop rotation almost
Well,
in that
yeah,
way.
it's the same principle, right? But the problem with perennial pastures is you're not rotating them most of the time, right? Like, no, some people do, mind you, but the average person probably wants this, you know, ideally they'd seed a pasture and then not have to worry about reseeding it or anything for
Mm-hmm.
10, 20 years at least, right? And so if you can't, you know, the problem is, I think, as well as what we frequently see is that kind of the density of legumes and alfalfa in particular declines over time in a lot of those systems. So you want to kind of reintroduce that legume component to get that nitrogen fixation and increase the forage quality. And, it's, you could potentially just completely, take it out of rotation as a pasture for a year and then seed it to annuals, and that, that kind of rotation certainly would work to kind of beat down some of those alfalfa specific or for legume specific pathogens in those systems. But if you've got alfalfa that's been growing there for 8 years, it's not really the rotation frequency if you consider the life cycle of a pathogen, right? Like it's depending on what it is, it could, the population size of the pathogen could double in a day. And that's not compared to the lifespan of a perennial pasture. It's really easy for those things that kind of like grow and propagate and spread throughout the soil. And you're not going to be able to kind of like turn it over as quickly with perennial plants. Um, so yeah, so the rotation just isn't necessarily as easy of an option in that scenario.
No, and there's balancing what maybe is best for the soil itself and what's best for the operation are sometimes two different things.
For sure.
So, when we're talking about some of these areas as well that you're using inoculants in, anything you've done with salinity tolerance? Like, I know some of these pastures have huge amounts of saline areas.
Yeah. So for the salinity, we did culture. So one of the things we've been focusing on for salinity are those mycorrhizal fungi that I was talking about earlier. And we, at least my hypothesis going into a lot of this was that we've got a lot of saline grasslands out there already that have kind of like species that are fairly mycorrhiza dependent. And so that means that they interact strongly with those fungi. So they rely on those fungi to provide them nutrients. And so that environment should have mycorrhizal fungi or other microbes as well, potentially, that are really well adapted to that particular environment and may be highly beneficial for plants in that particular environment. Because it's not like saline grasslands have no plants, unless it's really salty, obviously. But um, most of the time, you still have uh, plants that are growing within these environments in natural conditions. And so, what happens when you've repeatedly cultivated an area over time is a lot of these things, especially the mycorrhizal fungi, because they tend to get lost from those systems. And you can think of the fungi like it's basically a big network of small strands of hyphae spread throughout the soil, right? They're located on the plant, but you think of it as like almost like a really fine kind of like spider webs spread through the soil pores, gathering nutrients. And so obviously if you're tilling that or disturbing that soil, that could cause some pretty strong changes or disruption of those fungi. And also the same thing, like they generally don't tend to do as well in high nutrient environments either. And so when you fertilize a lot, it kind of, the plants don't need the fungus as much. And so there's not as much allocation to the fungus. And as a consequence, their populations can decline as well. And so for saline lands, especially seeded saline lands, um, you know, our thought is that most of these microbes are probably lost from those systems. At least the beneficial mycorrhizal fungi probably would be. And so we figured, well, what if we culture the microbes from saline grasslands and then see whether or not they can improve salinity tolerance for a few different forage species? And so we did this for, I think, 31 different grasslands across Saskatchewan and Alberta, where we went out and found saline and non-saline areas within the same site, and then collected soil and cultured those fungi in the greenhouse, and then inoculated plants in the greenhouse again with those same fungi to see whether or not it could increase salinity tolerance. And we did this with both barley and alfalfa. Um, with alfalfa, the inoculations didn't really have as much of an effect as we thought it might, but we could talk about this a bit later. But alfalfa really is super variable in terms of like how different cultivars or genotypes interact with those fungi. And so we feel like we probably just picked a bad cultivar to use in that trial because we weren't well, we knew there was some range, but we thought this one did okay, but it turns out it doesn't really. So anyway, the alfalfa didn't work all that well, but we found some pretty good results in the greenhouse anyway with barley. And so in our initial trial, we had, close to a tripling of growth in saline soils for some barley with some of the inoculants. And so we thought, like, this is definitely something worth pursuing. And so we've really been focused on barley kind of in that, inoculation of saline grassland microbiomes going forward. But, there's been some challenges with it. But we're still optimistic and, I still think there could be benefit. It's just we need to kind of better define precisely when it's going to be beneficial and do a bit more work to kind of before we can really make specific recommendations on that.
So I know it's not super black and white here, and nothing in research ever is. Um, but when we're looking at cultivars, would you say there's specific cultivars for a specific plant then? Like when you were looking at the alfalfa versus the barley?
Yeah, I know I think cultivar selection is huge, right? And it goes back to the same thing I was talking about. Like, certainly some plant species are more responsive. I was actually super surprised that barley responded positively to inoculation. Because usually annual cool season grasses aren't exactly that, they don't benefit the most compared to other plants. Like some of the warm season grasses seem to do a lot better with mycorrhizas, but yeah, for barley in particular, we didn't expect to see much there, but it did, obviously. And then for like alfalfa, when I was talking about the rejuvenation stuff, like, some of them big increase, some of them big decrease, right? And so like cultivar selection, I think is huge. With the barley, we've been working at 3 different cultivars, all produced here at the University of Saskatchewan, CDC, Austinson, uh, Renegade, and Maverick. And what we're finding is actually, at least for our field trials and for the greenhouse trials that we've run too, is that even these barley varieties are responding differently to different microbiomes. And so, we're really trying to narrow in on why, and we're, trying to do a bit more work to get more kind of like mechanistic around that particular response, but we don't quite know why the varieties are responding so differently. But they certainly are.
When you're looking at the different fields you looked at, specifically, like when you're looking across, how much did the actual field measurements play into this? Like when you're looking small scale, larger scale, or et cetera, et cetera. Does that make sense?
Yeah, and so there's 2 different ways. Like stuff in the field obviously is super important. Like, you've got microbes, the world for an individual microbe, its world is really small compared to, like how we perceive the world. And so some small level fluctuations within a field, like, tops, bottoms of hills, hill slopes, things like that are going to be like, that's a different planet basically for a microbe going from the top to the bottom of the hill, um, just because of all of the differences that you're getting in terms of like moisture retention and things like that. And so, field measurements are, when you're trying to evaluate these things, I think field measurements are incredibly important. And, we really need to do, I think we have a fair bit of work to do yet to kind of identify precisely when everything is going to work. But even like from inoculum development or identifying where to pick the microbes, like we're still, it's something that we're working on, and hopefully we'll have an answer for that one sooner than everything else. But, we know that some of the inoculants were more effective on average. And so, and I think a lot of that probably has something to do with which plants were actually located within the fields where we collected the inoculums. And so we just haven't had a chance yet to kind of really tease apart what's driving, like where you can find soils that would yield a good inoculant. And also, we don't know quite kind of like where those inoculants are going to work best. Ideally, in an ideal world, you've got something that works everywhere, but I, it's not ideal out there. So
Conditions aren't even the same on the same field every single year, so it's very
no,
difficult.
it's super like, you could say all you want about like different cultivars even being better for rejuvenation, but if it doesn't rain, it doesn't matter.
Yeah, yeah.
And so, like, and it's the same thing. Microbes are just as responsive, or probably even more responsive to variation in precipitation. So
Have you done any work with, low nutrient or drought conditions?
we have actually. So one of the other avenues that we're pursuing with a lot of this is we are trying to identify alfalfa breeding populations or cultivars, and also with sandpoint as well. Trying to identify, we're working with a local forage breeder here, um, using some of his breeding populations for these species to try and identify, uh, you know, breeding populations that do really benefit from mycorrhizas. And so we went out to a couple of his fields where he had a population of alfalfa, like a breeding trial for alfalfa and another breeding trial for sandpoint. And we collected plants from those fields that varied in size and then brought them back to the greenhouse and then transplanted them into pots in the greenhouse and let them establish. And so what we were curious about is whether or not those plants that were really heavily colonized in the field, whether or not they'd be more or less responsive to drought. So when we brought them back, we dug the soil out from underneath them and repotted them in the exact same soil that they were growing in before. And like I said, we let them establish. And then we set them into three treatments. One was um, just good growing conditions in the greenhouse. They were fertilized and watered regularly. And then a second treatment was a drought treatment, and then another was a low nutrients treatment where we didn't actually fertilize them at all after we put them in the pots. So just whatever nutrients already happened to be in the soil they were in. And so what we found with that actually for both alfalfa and sandpoint is that those plants that were more heavily colonized in the field did a lot better under drought actually than those plants that weren't. And
Interesting.
so we found a pretty strong positive response. And I think I can't remember which species was which, but for one of them, it just grew a lot better under the drought conditions. And the other one, it actually was able to reduce the amount that it died back. And so less of the shoots that it produced died back under drought conditions. So it was able to just kind of like maintain its biomass, green and, from a cow's perspective, nutritious rather than dry straw. And so so the drought really, we saw a very strong response from heavily colonized plants there. So suggesting that, at least for those, what we think is that those cultivars or those breeding populations that really, do get heavily colonized are actually capable of better surviving drought conditions than those that are not. And we do know from some additional work we've done, kind of like breeding heavily colonized plants together or crossing them at least, heavily colonized plants and then seeding them out into the field again. We do know that heavily colonized plants generally tend to produce heavily colonized offspring. So we think it is heritable. We haven't done enough work in that area to say whether or not it's something we can easily select for in a breeding population of either of these forage legumes, but it certainly shows some sign of heritability. And so, we think that could be important, is selecting those particular forage legumes that might interact strongly with mycorrhizas and therefore be much more tolerant of a fluctuating environment. We saw similar benefits, so low nutrient, but not kind of to the same extent. And so, again, I'm having trouble remembering precisely which species it was, but for one of the species anyway, we did see similar benefits under low nutrient conditions, but not for the other. But certainly drought, it was consistent across forage legumes.
Which is definitely a positive news story because that's exactly our conditions seem to be changing more and more every year, and drought seems to be happening more and more often than it maybe used to.
Yeah, for sure.
So anything we can do to kind of keep that those pastures growing absolutely is a positive. How far are we off from being able to use some of these inoculants in the field?
Yeah, the inoculants I think are maybe a little bit further off than some of the other things that we're working on. In particular, just because they've shown such site to site variability. And I know, there's lots of inoculant companies out there that sell a single inoculant for one crop and they say, this is the inoculant for this crop, just use it. And then, like 50% of the time, it doesn't even establish. And so, I don't ever want to be the person that promotes something as going to be super, ask somebody to go out and buy something that I don't know is going to work. And so I think we really like, from the inoculum perspective, I think it's more, I'd liken it more to kind of like it would be more successful if it's more boutique. And so, for a specific set of conditions, this inoculant works. Makes it a lot more difficult to kind of mass produce these things, obviously. And so I do think that's a little bit further off. Um, Unless, maybe we can find, we're doing a lot of, I've got a postdoc who's really keen and he's doing a lot of, or he's planning on doing a lot of genomic work to kind of look to see on what the molecular interactions are within the barley roots, assuming again, we can find financing for it. But I think if we can unlock that kind of, a detailed understanding of what's going on, maybe it'd be a little less trial and error and you could select more, varieties that we know are going to be responsive to specific inoculants. But developing a universal inoculant that's going to work for every situation is probably, maybe it will. Maybe you can do it, but I think ultimately those are going to be a lot more complex than the inoculants that are currently on the market, because you're going to need microbes that are adapted to kind of like drier conditions, wetter conditions, saltier conditions, less salty conditions. And so getting things to that point is more challenging. I think, there's lots of those that people can do still, even if it's not purely inoculant, and kind of like, even for like, for example, we collected the soil out of that breeding nursery where we collected those same plants. We cultured the microbes from that breeding nursery as well. And then we used that to inoculate the plants. And actually, like, barley did pretty good with that particular, with the legume breeding nursery inoculant. Um, as long as it wasn't saline. It did quite poorly, that particular inoculant under saline conditions. But I think what this says, though, is that if people can manage the land to kind of increase the abundance of these beneficial microorganisms, like inoculants might not even be necessary in the long run. It's just about kind of like, conscious management of kind of plant micro interactions in those systems.
Understanding that there's a lot more going on underneath the surface than you realize.
Yeah, or knowing that, for example, like when we culture the mycorrhizas in the greenhouse, we use either sorghum Sudan grass or big blue stem, both of which are grasses that can grow in the field here.
Mm-hmm.
Right? Big blue stem might be a little bit more challenging in the super dry environments. It tends to be more of a kind of tall grass prairie species, but for people with a little bit more precipitation, like sorghum Sudan grass grows okay. It does produce some, if you harvest it the wrong time, it's not the best, but it produces some compounds that can negatively impact cattle. But as long as you manage it correctly, it can be actually quite productive, nutritious forage. And so, but just putting some of that into the system, maybe that increases it. Like we haven't done the work specifically to do it, but I know other people in other systems have, been try to plant what they call essentially nurse plants within those systems. And those are plant species that are able to kind of like culture the right microbes to enable other species to kind of come in and colonize and do better. And so, there are little things that may very well work. That might be a simpler solution than developing an inoculum. But that being said, an inoculum is, for barley grain producer or somebody developing feed barley probably aren't, in those scenarios, kind of like you don't want a complex forage mix. And so you're not, um, your options were a little bit more limited there.
Yeah, absolutely. And I know this is going to be a huge question here, but you're working on tons of stuff right now. Is there anything you would like to, before we wrap up here today, anything you'd like to highlight on some of the work you're doing, or I guess any messages you would like to send to producers?
You know, I don't know if I've got anything I really, I think we're taking some really good initial steps on trying to identify ways that we can manage forage systems to improve microbiomes. And so I think, we're coming up with some good ideas right now. And, I do think that, ultimately what I want to see is for us to be able to put the power back in the producers' hands to give them tools to be able to manage their own land effectively, whether or not they're buying anything from anybody. I know it's probably not going to be popular, at least not with people that sell things. But it's we're like lots of things I think people can do right away. Like right now, we're trying to do some work on increasing what happens when you increase genetic diversity within alfalfa, right? Like if you seed different cultivars at the same time, ones that are maybe with complementary microbiomes. I think there's lots of ideas out there that wouldn't necessarily increase the cost for producers. They're just kind of like change a little bit of like even just how they're selecting seed. And so I think these things are coming. I think, obviously when you work in a perennial plant system, it takes a few years for really be able to make any concrete conclusions. And, I think we've really only taken the initial steps in my group. But, we should be able to come up with, we do have, I should have looked this up before I talked to you today, but, we do know what the lineage was of the alfalfas that did well with mycorrhizas. You know, uh, we might be able to identify from that, like commercially available cultivars that are closely related to those ones. Right. And so I think there's some kind of like very short steps, and hopefully we'll have an answer for that within a year. But, other things, especially around inoculum development, that takes a fair bit of time. And then when you start talking about actually breeding mycorrhiza dependent or mycorrhiza exploitative um, forage species, I think that and it just takes quite some time to be able to breed forage cultivar. And so I think breeders have their work ahead of them for that one. But I think we probably have options out there existing. It's just about doing the work to figure out which ones they are.
And if producers are feeling really keen, is this research available publicly?
So the initial cultivar inoculation should be a report and also like a quick fact sheet put out by a BCRC, the Beef Cattle Research Council, on that one. And the Alberta beef producers have gotten reports from me on some of the inoculation work and the selecting for specific alfalfa and sainfoin cultivars or genotypes that are mycorrhiza dependent. And I'm honestly not sure where those reports end up or if they're publicly available.
They are publicly available on our website. So, yeah, absolutely. That's one place they can go.
Yeah. And so, but, we haven't, most of this is not published yet. So it's not in the scientific literature quite yet. Some of it will be soon. But we are kind of, we're working towards kind of trying to get that out there. But I think, like the lovely funding agencies like the Alberta Beef Producers, the Saskatchewan Cattlemen's Association, the Saskatchewan Agricultural Development Fund, and the Beef Cattle Research Council, who have been financing a lot of our work and are new partners through like Western Grains Research Foundation, Sask Barley, and the Manitoba Crop Alliance, who've been out, supporting some of our barley work. We report to them annually with the results of what we've been finding. And so, even in its preliminary stage, I think some of those things might be available out there through funding agencies, which are often producer groups. And so they are they're producer groups, so they probably answer to the producers. At least I'd hope they do. And so, that kind of the producers are really the people financing a lot of this research. And so a lot of it should be available through those agencies.
Absolutely. Okay, well, I appreciate all your information here today. It's been really fascinating learning all about microbes and everything going on below the surface of the soil.
All right. Thanks, Kara.
Thanks again for tuning into another episode of the Bovine Podcast. As always, if you have any questions or feedback, don't hesitate to reach out either through ABP's social channels, or you can reach me on my email, karam@albertabeef.org. Until next time, stay safe, enjoy some sunshine, and don't forget to check in on your neighbors. Bye for now.